Hello, my name is Taylor Reed. My major is forensic science and my mentor is Dr. Jody Barta, and I will be discussing DNA determination of ancestry of human skeletal remains from unknown sources used as teaching specimens. So the purpose of this experimental research is to establish if it is possible to determine the ancestry of human skeletons that are treated for use as teaching specimens through DNA extraction, amplification, and sequencing. So ancient DNA from human remains has been studied as early as 1985, where DNA was successfully extracted from 2,400-year-old soft tissue. And shortly thereafter, in 1989, DNA was successfully amplified from human skeletal remains. These ancient DNA studies help to understand migration patterns and identify diseases. So there are a variety of methods that are used to prepare and preserve skeletal remains. The first was prepared by Andreas Vesalius back in 1543. And some of the methods that have been used since then include consumption of soft tissues by insects, maceration, or chemical use. Mitochondrial DNA has been the focus of genetic studies in the 1980s to the 1990s. It was the first genetic marker to be used in the analysis of ancient DNA samples. This is because mitochondrial DNA contains a specific non-coding region known as the D-loop region that is known to contain very high polymorphic rates, and this helps determine the haplotype and the haplogroup for those polymorphisms. So the samples were chosen from the Clay Adams collection of skeletal remains. With further research, it was suspected that these remains came from the Bengal famine in India from 1943. This was because the chief supplier was from Calcutta, India at that time. To choose the samples, 11 femurs were lined up from largest to smallest, and the largest size, which will be sample 1, the smallest size, sample 2, and the medium-sized femur, sample 3, was chosen. So bone powder was collected from each of those by drilling 4 small holes into the shaft for extraction. So the first step was to do the bone powder extraction. The DNA extraction of the 3 samples was performed using the Wizard Genomic DNA Purification Kit. Sample 2 was also subjected to a PDQX extraction because there was a remaining pellet. So DNA extraction consists of 3 main steps, which is lysis, which breaks open the cells and releases the DNA, precipitation, which separates the DNA from any cellular debris, and purification, which removes any unwanted material or debris. The next step was to do an inhibition test. This inhibition test was performed to determine if there were any inhibitors in polymerase chain reaction that had blocked the Taq polymerase from extending the sequence further. Next, resilica was performed. This was to repurify the extracts. And then finally, rescue PCR was performed. This was done by increasing all reagents by 25%, which decreased the amount of deionized water that was added. This is to improve amplification. So PCR is one of the main methods that is used in this experimental research. It is used in amplification, and it produces millions of copies of the DNA fragment of interest. It involves 3 main steps: denaturation, annealing, and extension. During denaturation, the double-stranded DNA is heated to approximately 94 degrees, and this separates the double strand into 2 single strands. During annealing, the temperature is lowered to approximately 55 degrees, and this allows the primers to attach to the complementary DNA sequence of interest. And then extension is where the temperature is raised to 72 degrees. This allows the Taq polymerase to add the complementary bases to extend the DNA. This is repeated 25 to 40 times, and then it is visualized on an agarose gel. So Sanger sequencing is a sequencing method that was used in this research. So to do a little bit of an overview on this, the deoxynucleotide is fluorescently labeled, and it displays a different color for each base. So adenine is green, guanine is shown as, is fluorescently labeled yellow, but generally shows up black. Cytosine is blue, and thymine is red. So each fluorescently labeled deoxynucleotide is ran through a capillary, and a laser detects the nucleotide and transmits it into a sequence that is seen on an electropherogram. So the initial wizard extraction did not show a successful amplification for the DNA samples. This was suspected because DNA does contain a very low copy number. Therefore, an inhibitor test was performed to reveal that the polymerase was not blocked during a polymerase chain reaction, which is indicated by the bands that showed up on the gel. So Resilica PCR was performed. Not pictured is the first one, where it resulted in a successful amplification of RE-1, which would be the sample 2's PDQX extraction. Then Resilica was performed with 2 different primer sets with bovine serum albumin added. The gel exhibited primer dimers and a very large amount of smearing, which is seen pictured here. So rescue PCR was performed. There we go. Rescue PCR was performed and was successful for samples 1A, RE-1, and RE-2. RE-2 would be the second Resilica of the PDQX sample to extraction. Without bovine serum albumin, which is known as BSA, and samples 1A and 3A with bovine serum albumin for primers forward 16131 and reverse primer 16251, which is shown in the image here. Rescue PCR was also performed again with forward primer 16232 and reverse primer 16355, and it showed successful amplification for RE-1, which is seen in lane 11. So the samples that were able to be successfully sequenced were rescue PCR samples 1A and RE-2 with forward 16131 primer and reverse primer 16251. This is depicted in the top right image where it is aligned to the Cambridge reference sequence. In the bottom right image, rescue PCR samples 1A, 2A, 3A, and RE-2 successfully amplified and sequence with reverse primer 00018, and this was aligned here to the Cambridge reference sequence. There were no polymorphisms detected for these ones or the ones depicted in the top right. However, in the left image, sample RE-2 exhibits a base change at position 16269. This base change is from adenine to thymine. However, because this is not a common polymorphism, it could be a damage in the template DNA that was amplified, and further sequencing would need to be done to analyze that further. So in conclusion, it was determined that DNA can be successfully extracted, amplified, and sequenced from skeletal remains from unknown sources that are used as teaching specimens. And further analysis may include conducting PCR with more primer sets to sequence the DNA and detect any more potential polymorphisms. I would like to thank Dr. Jody Barta and the Madonna University Forensic Research Facility for assisting me on this research project and giving me the opportunity to work with these amazing specimens. Thank you, and let me know if you have any questions.